Hi, this is Mr. Warchi again, and this is the third screencast of the genetics unit. This is the last slide that you took a look at in the second screencast. Remember, if you have any questions, view the video over again, or you can read your textbook, or you can ask me tomorrow. So modern Mendel 5, incomplete penetrance. When an individual might have the abnormal gene, but they might not express it. For example, polydactyly, which means that you have too many fingers and toes. Count those fingers carefully. Okay. So people can have the gene for polydactyly, but not express it and pass those genes on to their kids, and they can have extra digits. Now, there are things called sex-limited or sex-influenced traits. So certain traits are associated with gender, with the parts. For example, my mother had uterine cancer. Now, I could carry the gene for uterine cancer, but I will not have uterine cancer because I do not have a uterus. Right? Same thing with prostate cancer for females, or for individuals, I apologize. So they are associated with the specific parts. Variable age of onset. This happens later on in life, like male pattern baldness. People aren't born bald. They go bald over a period of time, like me. Huntington disease. It doesn't happen right away. It waits until usually you have after you have kids, which is the sadness of Huntington disease, which we'll talk about later. So you have the genes, but you express the trait later on in life. Variable age of onset. The genetic of sex, gender. In 1910, Thomas Hunt Morgan, he started to work with fruit flies, and the stuff that he did with fruit flies and that stuff that his students did with fruit flies was simply amazing. Amazing. So he worked with mutations. White eyes was the first one. He found that a trait was associated with the X chromosome and would happen more in males than females. He discovered that males and females had two gender chromosomes. Females were XX and males were XY. Notice that males have an X and a Y, and the Phi individuals have two X chromosomes. The sex chromosomes or gender chromosomes are down here. If you're male, you have XY. If you're a Phi individual, you have two X chromosomes. Look how small that Y chromosome is. It's really kind of useless. It has just a few genes on it. So the X and Y behave like genes. They follow the law of segregation, right? So if you have X and an X, two gametes will form, and each gamete will get an X chromosome. If you have an X and a Y, two gametes will form, and each one gets an X and the other gets a Y. On the X is the DAX gene, which forms female parts, the DAX gene. On the Y, the SRY gene, or you can call it SARI, forms the male parts, or the penis. Right? In case of a tie, Y wins. I'm sorry. Right? So the SAR gene is expressed. So on the Y chromosome, we have the SAR gene. You have gene for hairy ears. You get other small set of genes, but it's just really kind of a small, tiny chromosome. And that's pretty much it. So Y equals penis, right? So because it has the SRY gene. So X equals female parts. So the X-linked traits or sex-linked traits, I call them X-linked because they're on the X chromosome, are on the X chromosome and they are recessive. X chromosome recessive. X chromosome recessive. X-linked traits on the X chromosome and they are recessive. Dang it. So some examples. Here are 4 examples of common X-linked traits. For example, color blindness. And we're specifically talking about red-green color blindness. These individuals cannot distinguish between red and green. It looks like it's gray to them. That's why they have those little colorblind test things. Yeah. Male pattern baldness. There's a specific type of baldness. When you have the sides racing towards the back and you have that bald spot, you have the ring of hair, which is really not a very attractive look. I don't think it's an attractive look. Hemophilia, when you don't produce the protein factor 8, and that's important in blood clotting. Duchenne's muscular dystrophy, when you're dystrophin, the protein in your muscles break down. Again, they're recessive and they're on the X chromosome. So Punnett squares with X and Y chromosome, they behave the same way. On top here we have the male. On the side we have the phi individual. The individuals give an X. Males either give an X or a Y. So you have XX, phi individuals, XY, males. Right? Makes sense, I hope. Who determines the gender of the child? The male does. Who did King Henry VIII kill when he couldn't have a male heir? His wife. That doesn't make sense. So a woman who is a carrier for hemophilia, right, marries a normal male. So I put over here the H's on top of the X chromosome so that you know, as superscripts, that they're associated with the X. There's nothing on the Y except for the SRY gene, but we don't care about that right now. Now, phi individuals could have either two dominants, right? Or they can be heterozygous up here, and no one would know until they have children. But if you're a carrier, you carry the recessive gene. So if I specifically say that you're a carrier, you carry the recessive gene. Males, on the other hand, you either have it or you don't. Either have a little h or a big H. If you have a big H, you're normal. If you have a little h, you have hemophilia. There is no such thing as a male carrier, and you don't put anything on the Y chromosome. I don't want to see anything on that Y. So in this case, the phi individual gives a big H, or the individual gives a little h. The male gives an big H, or the male gives a Y. So when you cross these things, we have two normal phi individuals. Two normal. Even though this is a carrier, you don't know she's a carrier. We have one normal male and one hemophiliac male. So if I'm bald, which I am, I don't blame my dad, I blame my mother. And who gave the bald gene to my mother? But her father. So my grandfather on my mother's side, if he's bald, I have a chance of being bald. Go back and start thinking about that. Because your dad doesn't give you the baldness, doesn't give you the X chromosome. Your dad, if you're a male, gives you the Y chromosome. If he gave you an X chromosome, you'd be a female or phi individual. So we can combine these with other traits. For example, AB, a male with AB blood marries a phi individual whose father was colorblind and has O blood. So if we're taking a look at a normal male, colorblindness, we're using B here. Might be a little confusing, but this is the colorblindness, normal, and AB blood, right? Over here, she's a carrier. Why? Because the father was colorblind and the dad had to give her an X chromosome. If dad gave her a Y chromosome, it'd be a boy. So she's a carrier, right? And has O blood. So we have to figure out the number of, or the types of gametes. Well, we have two heterozygotes here. So 2 to the second is 4. So we have 4 gametes. We write down 2 big B's and, right, and 2 Y's. And then we alternate A, B, A, B. Here we have 1 heterozygote. So it's 2 to the first, which is 2. So you have, right, X big B, X little b, and all of them get the O. So then we set up our big Punnett square like this. So we get here, we X big B, X big B, A, O. So we have a normal female with A blood over here. Now, with these things, we have to talk about gender, because we know the gender. We have X big B, X little b, A, O, normal female, A blood. Boom, right here. A normal female, B blood. Normal female, B blood. Right here. Normal male, A blood. Out here, colorblind male, A blood. Normal male, B blood. Colorblind male, B blood. X inactivation in phi individuals. Females have 2 X chromosomes, but during development, one of them shuts down. And if it condenses, it becomes a bar body. There are a couple of genes that are active on it, but it's pretty much shut down. It's located near the nuclear envelope. It's random on which one shuts down. So a phi individual is a mosaic between the mom's X chromosome and the dad's X chromosome being active. And it is activated, the second X chromosome is activated when you form gametes, interestingly enough. Chromosomal alterations. So we can alter the number and the structure of chromosomes. This isn't a good thing. These are usually bad things. So we don't want this to happen. So if you alter the number of chromosomes, You do it through non-disjunction, and you end up with something known as aneuploidy. We talked about that in meiosis. So if you have 2n, which is 46 plus 1, you will have 47 chromosomes. So trisomy 21, which is Down syndrome. Trisomy 18, which is Edwards syndrome. That is 2n plus 1, 47 chromosomes with autosomal. These are not the sex chromosomes. If you have 2n plus 1 with sex chromosomes, you either have too many X's, like XXX, triple X, XXY, which is Klinefelter's. It's a male because it has a Y chromosome, Y equals penis. XYY, which is Jacob syndrome. You can learn about that later. Or you can be missing an X sex chromosome, only survive if you're missing an X. So in this one, it's 2n minus 1, which is 45 chromosomes. This individual is XO, X and nothing. It's a female, individual, because there's no Y chromosome there, right? And it's called Turner syndrome. Or you could have problems with the chromosome structure, and this is done through crossing over. You can have deletions and insertions, also extra copies with unequal crossing over. Inversion, remember the piece of the chromosome can flip around and join. Breakage, it can break out, cross over, or start crossing over and just be lost. Or you can have a translocation where that piece that's lost bind or joins with a non-homologous chromosome. So you can alter the chromosome number as well as the chromosome structure. So when you have a baby, we want to take a look at genetics of the baby or the chromosomes of the baby. We have these prenatal tests. Now, the first prenatal test is an ultrasound in which you bounce sound waves off of the baby. The sound waves create an echo, and the computer makes a picture. And here's a little ultrasound right there, and there's a little baby. And these are the old school ultrasounds right here. You know, baby's head, baby's leg up here, the arms coming down here. And it was pretty good, you know, you could see a lot of things. But now they have these three-dimensional ultrasounds right here. It's kind of freaky. Look at the detail they get in those things. It's amazing. What else? What other prenatal tests do you have? Well, you have chorionic villus sampling. This happens from anywhere from 10 to 12 weeks. You take a piece of the placenta. Right, these are baby cells. You grow the cells. You look at the cell chromosomes. You look at the proteins they produce. Right there, and you do it with ultrasound, so you're not, you know, trying to take a piece of the baby. Or you take a piece of the placenta, that's baby cells. The amniocentesis is another prenatal test. You do this from week 15 to 20, second trimester. You go in and you remove some of the amniotic fluid in here, right? Because baby cells are being sloughed up all the time from the baby. You suck up the fluid, you suck up some cells, you grow the cells, and take a look at the chromosomes and proteins that the baby cells produce. You can grow the cells, you take a picture of them, and you can arrange them, and you can put them in order, and you get something known as a karyotype. We've talked about this before. This shouldn't be new. And we can have normal karyotypes. This is a normal male. How do we know it's male? XY. This is a normal female. How do we know? XY. You know there's nothing on the Y because there's no Y chromosome there. There are two X chromosomes. That's a phi individual. You can also have non-normal karyotypes. Right here we have trisomy 21, 321 first chromosomes. It's a male boy with 321 chromosomes. Or, you take a look here, what's wrong? Anybody see what's wrong? Oh, we only have one X. Only one X. All right, Turner. And we're going to stop there. So, if you have any questions, please review the video, or you can read your textbook, or you can ask me tomorrow. And have a good night.